Okay, should I start?
Yes, absolutely. I'll give you an introduction here. I just want to thank everyone for attending this month's Clinical and Translational Science Institute's Translational Science Seminar Series. Today, we are welcoming Dr. Keith Chang. Thank you so much for presenting your work here. Dr. Chang is a distinguished professor of pathology with joint appointments in biochemistry and molecular biology, pharmacology, and public health, a co-hire of the Institute for Computational and Data Sciences and lead of the Penn State Computational Phenomics initiative that involves faculty across Penn State, including IST, statistics, and CSE. His training includes a BA in biomedical sciences from Harvard, an MD from NYU, residency in pathology at Brigham and Women's Hospital and University of Washington in Seattle, and PhD from University of Washington and Fred Hutchinson Cancer Research Institute with Gerald Smith, studying the molecular genetic mechanisms of recombination, and then a postdoc with Larry Loeb, where he studied mechanisms of genomic instability. So thank you, Dr. Chang. I look forward to hearing your presentation for today.
Okay, thank you, everyone. So I would love if this talk made people happier after hearing this. And so I hope you'll be able to share my excitement with my recent lifelong goal of being able to see tissue in 3D, pathology in 3D. And I'll explain to you why I'm so excited about it, and hopefully you'll like some of the things we're doing and can see some potential uses of this technology for your work, or at least applications that might impact your work. So the idea of geometry of life and disease, as you'll see, is a theme that I realized could it ties into what biology is and utilizes that in order to help us learn more about the mechanisms of biology and disease, and there'll be translational impacts of this work. So another title I had used in Berkeley recently in Department of Energy is to explain this very interesting convoluted course of my science, and it inevitably leads to what I'm doing now. Actually, it's very, very interesting. Happy to have lunch with somebody someday if you want to talk about it in more detail. But I'm going to be covering briefly for fun how I got from cancer research to the study of skin color, and then that actually ties into my work in computational phenomics. So we'll talk about whole organism phenotyping, this term that I'll say it now, histotomography, which is a CT for small objects, but for all the tissues, so seeing all the cell types. And we're pursuing broader access of this technology by users across the United States first and the globe. And there's a lot of tool development and integration that's going to be going on with a grant that I'm submitting tomorrow. So cancer is a disease of somatic mutation, and what I did with that, this is the start in the my first projects, how I got going here. I asked, how do we model somatic mutation? And so we used a forward genetics approach, which consists of making mutants in whatever the process is that you're interested in. In my case, I was interested in the rate of somatic mutation and look for an assay where you could detect somatic mutation. So George Streisinger found this fish called golden and found that the embryos are transparent at day 2, you can see here. And if you cross them, you get heterozygotes. And if you treat those with gamma rays, which knocks out random genes, you can see patches of cells that are actually mutants, cellular somatic mutants in that same gene, so that the wild type allele. So it's mutant. And so I wanted to use this assay to detect what are called somatic mutators. There were traditional mutators done and looked at in bacteria, which result in our knowledge of, for example, Lynch syndrome in DNA repair. But this in long and short, I won't talk about this screen except to say that we got a nice article out of it, and it resulted in susceptibility to cancer. But something that raised my curiosity was why these are the adults of those same fish. and they're lighter in color. And I asked, well, why are they lighter? And if you go back to cell biology, the melanin pigment is contained in melanosomes. They're little lysosomal-like organelles that contain pigment in them. So there are only a few things that can happen to those. So there can be fewer of them. They could be smaller, and they could have less dense paint inside. So we looked at these mutants, and in fact, they have changes in actually all three. So this is wild type, this is mutant. There's fewer. They're smaller, actually, and they have more diffuse, the lighter pigmentation. So that's really odd, it turns out. So then I ran to the textbook of dermatology, and it turns out that the same three changes occur in light-skinned humans, and namely Europeans. And so then that raised this question, so odd, can a variant in that same orthologous gene in humans actually be responsible for a difference in skin color in human populations? Long and short of it is the answer is yes. And this is a lot of gobbledygook to say that we did positional cloning and found these genetic markers that were near this golden gene by genotyping a lot of mutants, one gene at a time, one marker at a time. It's actually quite laborious back then. And this piece of DNA contains this region. And so we injected that piece of DNA into golden mutants, and lo and behold, you get rescued cells. So that means we have the right region. Then we had to sequence the whole piece of DNA. None of those genes were candidate genes for pigmentation. And lo and behold, we ended up doing, after the pack rescue, as you saw, we have some rescue, but we actually rescued with the particular RNA for that SLC24A5 gene. So that is the first piece of evidence that we have that that's the right gene. And then we find a mutation in it that causes a nonsense termination codon in this gene. And if you look now at the human gene and ask, can it rescue, you inject the human RNA, and in fact, it does rescue the golden phenotype. You can see this is the golden mutant phenotype, and it actually darkens up with the human RNA, which means that there's a conservation of function across evolution. And if you look at the amino acid sequence of this 500 amino acid gene, around the region of this mutation, every single vertebrate sequence to that date was the same amino acid in this whole region, except for this one group, which is called Europeans, CEU, in the original HapMap populations. And if you look at the display at the time, which was these pie charts, you see one genotype for Europeans and another genotype altogether in the Chinese, Japanese, and Yoruban Africans. And this is a layout of the whole region with covering these other genes. And it's very odd that over 150 kilobases of DNA, that's all the same genotype, which means there's a selective pressure for that mutation. This is the mutation here. So if you look in the entire genome, because it could have been just, okay, well, maybe that's all over the place, but it turns out, no, this is the single largest region in the entire genome of only Europeans that is homozygous. So that indicates a very, very strong selection, and it ended up being a nice cover article. And we use Photoshop mutagenesis to color this hand to illustrate the dark pigment is in Africa. So at any rate, and this is the population distribution. And the next question is East Asians, and Dr. Casey Ang and I are chasing that gene. It's been over a decade, and we think we have a good idea what the gene might be, but that would be the next story. So in pathology, that's my training. I went through medical school and fell in love with pathology. And here is a one time in my life picture that I got of a squamous cell carcinoma, and there's a hexapolar mitosis. Now, why is this so odd? Why is it so rare? Well, it's because that cell is actually quite a large diameter. It might be 30, 40, 50 microns in diameter. And we happen to catch the plane over which the 6 poles of the mitosis are going, which is not normal. Of course, normally you have 2 poles. And so it's very rare that you would see this. And how could I make it so I could always detect these? because atypical mitoses are a typical feature of bad cancers, which is an illustration of their genetic variation. There's no two cells that are the genotypically or have the same chromosome number practically. Whoops, wrong way. So if you look at these cancer cells here, you can see how large they are. And we know that in our histology, we actually only look at 5 micron sections. So this is an illustration of one of those cells in the previous slide. And these are the actual thicknesses in scale of the slices that you see on a histology slide. And these are the representations you would get. And none of them are going to be fully telling you about the volumetric aspects of that cell. And so really what you need is a 3-dimensional histology. So how do you get there? And this is just another illustration. Many of you have seen Sagrada Familia in Barcelona. And if you took a 1-inch slice through this, how much information would you really get about the details of the inside of this church? So you would get more if you have a 3D image, wouldn't you? So similarly here, this is prostate cancer, the most common cancer in males. And we typically grade these, we have the Gleason score, and you compare with these ink drawings, hand-drawn ink drawings of glands and their patterns. And you can see in this one slice of a cancer, where the cancer is up here, that there are many morphologies here. And really what you want to do is look at the entire gland, or at least your entire specimen, and then assess what the worst part is, because that determines your prognosis. But we really can't do that. And here you see, say, a circle. To what extent is that a bubble, or is that a tube or part of a gland structure? So wouldn't it be nice to have a 3-dimensional structure of these glands? So now let's turn to another aspect of this kind of imaging. We often look at model systems. For drug development, you need to find out what the responsiveness is to these drugs, or toxicity. That's the reason that 4,999 out of 5,000 drugs are not used when you have drug candidates. You need to reason it's nearly $5 billion, can be $5 billion to produce one drug, is because most of them fail. And early failure is really important. So it'd be really important when you use model systems to really make good use of these animals, which are live animals. You don't want to waste them. So it'd be good to look at the entire thing and get complete phenotype. The other problem with phenotyping is that, at least for non-pathologists, there's no lab that has complete expertise. And the poor graduate student thrown at a project to read the histology slide, and there are recent illustrations in the literature just published in science, the likes of Science, that are completely bogus because they don't actually know what they're looking at, and they don't share the whole image. They don't share the whole organism image because they can't. So how do we get complete phenotyping and allow people to be able to validate the results or to check the results? And what are the barriers to this complete phenotyping? I already told you how relevant it is in terms of the drug development. It's also relevant if you get for example, a lymph node dissection, and you want to detect the cancer. And if you miss the cancer, and they might be only a few cells, and I had one case where I saw literally one melanoma cell in 13 lymph nodes, I could easily have missed it. If you could have 3D histology, you could actually detect every single one and count them and actually measure their features as well. Imagine, by the way, in that 50 micron cancer cell, having not just one 2D angle to look. When you're looking from the top, you have great resolution XY, but Z-axis, you have no resolution. Instead, if you had 50 micron cell, you could have 100 slices at a half a micron. That's a lot more information. So phenomics is the collection of all phenotypes in all organisms in all cells. And what a phenotype is, like what an ant, normal ant or a diseased ant or a human or a rabbit. And a computational phenomics is simply an expression that we want to use computers to do phenotyping if possible. As you probably know, when we do pathology, diagnostics, which is really key to our treatment of patients with cancer, for example, we're being a little subjective. We can only see a portion, and our minds are remarkably able to infer the three-dimensional structure, but there's no quantitative measurement. So we're not quantitative, and it is a little subjective, and we can miss stuff. So finally, the biggest rage of the day is spatial biology. Large-scale transcriptomic or proteomic or other omic molecular analyses of cells. But these studies are typically either they're not done with intact tissue, or if they're done with sort of intact tissue, it's slices. And again, you're going to be missing parts of the cells that you're slicing or the tissues. So, and if you look at the details, which we won't go into today, there's actually a lot of concerns. There are a lot of details to limit our ability to interpret and to limit the amount of data we can get out. It's not actually literally every single cubic micron that you're getting information. So I want to make a few definitions now for a moment. I want to pause and define pixels, which everybody knows about on your screens that you're looking at right now. It's divided into little squares called pixels, and they have dimensions that are x, y equal. And then the voxel is simply a three-dimensional pixel. So in fact, the pixel has a volume. So you're actually looking at a voxel from one side. But if you have say a CT scan, and if the voxels are isotropic, that is each axis is of equal dimension, you can rotate the thing and not get distortion. A lot of images, for example, a confocal imaging is commonly anisotropic because of the limited number of angles of projection you get. So what our goal is to have isotropic voxels so you don't have distortion. And I thought people talk about bit depth, and to maybe demystify that a little bit for those who aren't familiar with this, if you have only one bit to look at, it's either a 0 or 1. There's two values to look at. There's only two values possible. If you have two bits, say a pair of bits, then you could either have a 0 or 1 in each position. There's a total of 4 possibilities. So now if we extrapolate up, 65,000 values is pretty good. So I'm aiming at 16-bit imaging. 8-bit turns out, as you see, 256. Believe it or not, we can actually see more than 256 degrees of variation. It's meaningful for us. So I'm aiming at 16-bit. So there's your explanation of that part. And for resolution, there's optical resolution that we read about in physics, if we took physics, and using Rayleigh's equation, which inputs wavelength and numerical aperture of the lens, and that defines the size of the object you can discern. But in digital imaging, you have pixels, and so the pixel is the dimension that is half. When you take that optical resolution to see an object of unit 1, you need to have a pixel of half that size. That's a rule of thumb. It's very nice, very convenient to remember. And in terms of microanatomy and histopathology, which we use to diagnose tissue, make tissue diagnoses, you need to have empirically a resolution of 1 micron. So we need a pixel of a half a micron, at least, and to do pathology, do histopathology. And if we want to do it in 3D, we want it isotropic, as I said earlier. So how do we get to isotropic images with a half a micron voxel? How do you get, now the other thing is, how do you get us large enough size image in order to fit, for example, a whole model organism or an entire pathology specimen, which are commonly in the range of a centimeter or so. And so the way we do tomography, and this is the same principles as we use for CT scans, you take rotational projections, you don't spin the person. In the case of CT, you actually spin the X-ray source and the detector, which used to be the weight of a Honda Civic. It's not anymore. And in micro CT, instead we rotate the specimen when you have the source and a detector. So what you don't have to remember this, but what we output is an image stack calculated from a series of slices that are reconstructed from the whole series of projections, one row at a time. And so the other feature I want to emphasize we want to get is pancellularity, because you don't want, if you have a fluorescent marker, you may only see one cell type. Instead, in pathology, as you know, we stain for all cell types, and we can recognize them all by their morphology. And so the ideal here I'm chasing is to be able to do this in 3D. So we want high resolution and large field of view, and physicists will tell you can't have both. So I'm a little greedy, and we designed a new lens system, which instead of looking, maybe a couple inches tall and maybe, I don't know, 3/4 of an inch wide or an inch wide at the most, to something that's like several inches wide, or 3 or 4 inches wide, and like 50 pounds and 26 inches long. So that's actually what we have now. So the idea here is to be able to do whole organisms. We can study gene function by looking at all the phenotypes across all tissues. We can look at organisms, or any organism you can fit in association with climate change, toxicology, human impacts. There's drug toxicology, and there's, of course, tissue diagnosis that we use every day in the hospital. So our phenotype, what we look like, our state is defined by our genes, our environment, and by disease. And these phenotypes can include, as we've just been discussing, tissue structure, as well as physiology, or how fast your heart beats, et cetera. Descriptions, down to the descriptions of cells and their relationships between each other. How many are there, what the proportions are, how close they are to each other, what their shapes are, et cetera. And then there's now molecular phenotyping. And so how do we make this computational? And how do we defeat the problem of limited expertise and get complete? So in phenomics, I told you mutants, we have mutants caused by gene change and exposures, and we have disease, and those give you genetic, environmental, and disease phenomics. And there are all these model organisms that we use to study phenotype, looking at genes, disease, and environment. And the phenotypes can be in different organ systems, cells, and at different life stages. So how do we make, this is a pretty big call, right? So how do we reduce this confusion, mass confusion? How do we feel a sense of hope instead of hopelessness? So we have to realize that the unity of nature creates feasibility. Every living thing, it turns out, it conveniently so, is comprised of cells. And if it's a multicellular organism, those cells are organized into tissues and organisms of similar structure, not often different, but there are some similarity up and down phylogeny. Of course, different in plants and animals. Every disease, it turns out, as a pathologist will know, can tell you, is associated with three-dimensional changes in tissues. They change cells, they change how the cells are organized with each other. Let's see what time it is. Good. And the molecules, of course, are similar, and they have spatial specificity. They're in specific cells, or in even specific cell parts of cells and organelles. And they can also be in the fluid, in the intercellular spaces, for example, and into structures like cartilage or bone. Tendons and bone. And in terms of imaging, everything we're looking at, the cells and our organisms, us as organisms, are all 3D. So we want to be able to get their 3D phenotypes. We want the imaging to be unbiased, not biased. So ideally, we can utilize the biased phenotyping for specific molecules, but also have availability of unbiased phenotyping, because only then do we get a truth without assuming things ahead of time. All right, so we also, and I think for those of you familiar with glass slides, I think all of you are, when you look at tissue, we want to get to a centimeter field of view if possible. All right, so here's the aspiration. We want to define the geometry of life and disease. We want to characterize, ideally, this is not a one-person project. Every cell, every tissue, every organism, and every life stage in health and disease. Now, fortunately, there is a finite number of cells and tissues, and yes, there are many organisms, but it's a finite number, and the changes that occur actually fall into classes. That's why a comparative pathologist can look at a slide of anything. That's why I can interpret pathology in a zebrafish, even though I was trained in human pathology. So we've got this framework of life, and all these are parts of a house. And of course, you can substitute all of your molecules and phenotypes on that. And so these are more challenges. We have world populations and many organisms. We have limited expertise. We need everything to be visible over millimeters or centimeters, which has not been possible, even with the best of equipment when I started. And the file sizes are really huge, can be tens of gigabytes. And the tools are really hard. Your computer can't even open, a terabyte size image. Fancy computer can, but our typical laptops can't. So how do you deal with that? How do we even look at the samples? How do we look at the images? And if you've got a phenome project that taxpayers' dollars are spent on, wouldn't it be better to have the organism looked at with today's technology and put it away and look at it in tomorrow's technology later on? We need to make images available to people so that bioinformaticians and others can go play with them, discover more, validate results, and develop tools for looking and analyzing the information. So this is, these mechanisms need to be available across organisms. And believe it or not, this is actually all possible. So we have the first practical form of 3D histology created with a Penn State-led team here. And we're developing, as you've seen, this idea of computational phenomics, where we can define characteristics of cells and tissues. And we actually are also known for creating a zebrafish and now a Daphnia atlas. And the tools that we develop for the atlasing can be applied to any atlas in any organism. And what I'm trying to do in the remainder of my career is to create resources that can be used as broadly as possible and to share them as much as possible. We want to help society use facts and science to benefit society. So I'll cover briefly here then the technology. You don't have to know every element here. There's fish. This is a dime. You know what a dime is. And this is how big the juveniles were that we looked at in the larvae. These are about the size of a needle biopsy here, the width, maybe a little smaller. And so we take there's a synchrotron, there's a kilometer diameter synchrotron at Argonne, and all their fancy equipment allows you to produce relatively monochromatic X-rays that are projected in parallel geometry through our object, our fish. In this case, we've done a lot of other things too now. And then it shines on a scintillator which glows, and the glow is captured by a lens system that is now used to be just a microscope lens and is now our custom system. And it bounces off a mirror, so you don't actually X-ray your machine, your camera. And then you collect images as the sample rotates. So you can, typical experiment, you will rotate this thing, take a picture every 0.12 degrees and have 1,500 pictures resulting. And this is the images you can get out of it. So this is from our 2019 paper in eLife. It's easy to find. Just Ding or Chang and then zebrafish and histotomography or eLife. And you can see all the cell types. So this is the eye with all the retinal layers, photoreceptors, and the different inner and outer granular layers. This is cartilage here, blood cells that we've just submitted a paper to eLife actually describing the geometry of those cells and finding them in the entire larva. This is the so-called pneumatic duct. Here's the air bladder, And what air gets in there through the pneumatic duct. I guess they can swallow air and it goes in there. And these are actually the striated muscle that surround the air bladder that you can see detail. And this is histology, real histology on our atlas. And this is a corresponding area of a different fish, where you can actually, as you'll see in a minute, detect the three-dimensional structure. So these are what we get in histology. You can look at those all day, but you wouldn't know this is what it looks like. And this is of even lower resolution than this, but you can see the three-dimensional structure now. So this gives you an idea what 3D can do for you that 2D cannot. This is actually pretty low resolution image. It's a 1.4 micron resolution of a whole juvenile. And what we're doing is asking the question, addressing the question, can you see cell features looking at the surfaces of, say, the gill or the gill cavity? And in fact, you see little bumps on the leaves of the gills, and those are actually nuclei poking out, pushing out from inside. This is from the larva from that paper, and what we were able to do is to segment out individual nuclei first, and then we're able to cross-register with a published atlas and label each of the nuclei as to where it is, and assign regions to the nuclei there. But the more fun experiment was this one, where we actually asked the question, can we look at the distribution, the density of nuclei? So how many neighbors do you have on a per nucleus basis? And if you have a lot of neighbors, you color them red. And if you have few neighbors, you make them blue. And you can see, this is a shocking result. We have 5 siblings, same parents, born the same day, if you will, fertilized the same day. And they're at the same age, and they all look a little different, which is not too surprising considering that 5 siblings of a human family never look the same, do they? and here we can see that we can get that level of detail in these individual fish. A little frightening because you often do experiments in which you will divide a set of siblings up into groups, and you can imagine with any variable you like, you can probably find some variable given this variation that correlates with your variation that's actually not going to be real. So this is work that Spencer Katz, who just gave the talk at the graduation a week ago, this is his work where he's able to stain pigment cells in the whole zebrafish. And this is the work we were just sent in, and we're able to segment out manually first and use machine learning to find remaining blood cells and then characterize them geometrically. And we're going to be pressing on to look at other cell types. And it's going to require, of course, funding to get that work done. And this is just, each sort of 4 different cells that we have there. And then we have each of the cells with statistics that we've been able to calculate with a lot of help from other scientists working with us at University Park, the Huang Lab, for example, in IST. And we're, of course, wanting to look at many organisms. There's Daphne on the bottom left. We have some nice pictures you'll see in a minute. And want to look at Drosophila, we can actually we're imagining looking at mutants for every single gene, which they have in the Bloomington Drosophila stock center and mouse embryos. So instead of the house, we're going to have these 3D structures of organisms on which we can add, we can put in a space-specific manner all these other omics. And there will be clinical relevance and global relevance to all of this work. So the idea is to use computational phenomics for validation and discovery. And then actually, if you read about happiness, which I am a student of, the sense of wonder is actually a high correlate with people who are happy. So I want to make people happy. That would be really nice. All these other fun terms, of course, will be forwarded, will be advanced as well. And so we're creating a geometry of life consortium, or geometry of life and disease if you're more NIH-y. And there will be standardized sample prep that'll be developed over time, that'll be published by each of the groups. And then we'll have a synchrotron micro-CT imaging resource that the grant that we're submitting is to get the last pieces together for high throughput work across the globe. And then there'll be Galaxy is something Penn State's known for bioinformatics, and we're going to be adding image bioinformatics to that is the idea, and integrate data across organisms and cell types and data sources, and different types of imaging as well, and different length scales. Okay, so we can, as people can imagine, any given chemical or gene mutation causes a series of changes across different organ systems that are specific. And I have called those phenotypic signatures. Other people have other names for them, but it's actually sets of phenotypes. For example, you might say flu causes cough, sneezing, and itchy, whatever. So that's a phenotypic signature of flu, for example. And so just extend that to phenotypes caused by gene mutations, for example. So this is what I call Papa Bear, Mama Bear, and Baby Bear. This is Baby Bear is what we used to use, these microscope lenses, what all the micro-CT machines, even in synchrotrons, utilized. And the field of view is about 1.7 millimeters maximum for a submicron voxel pixel. And for Mama Bear, we have 5 millimeters for a half a micron resolution. And then Papa Bear is the one that won't even fit at Berkeley. It fits in Argonne National Lab, and that has a 0.7 micron. That's a submicron, but 0.7 micron. It's similar resolution as the original publication, but now over a centimeter. And this is Max with that camera system in our hutch in here in Hershey. This is a figure that was just finished about an hour ago, and for our grant. And this is what we're targeting, is to get to a centimeter at submicron voxel resolution. And these are the other technologies that have been used before. None of them have either the field of view. If they have the resolution, the higher resolution, they don't have the field of view. And if we have the field of view, you don't have the resolution. So ours has a sweet spot here. And this is to illustrate what we can see this is the front of a frog, and this is the individual fields of the past camera system that they had. And this is for example, a nano CT machine. So you can see the vast difference in area you can detect. This is a reconstruction of that nose there. And we do have proof of resolution. And let's get to the fun movie here of a Daphnia. This is a 2 millimeter tall organism. And we're able to image this at a half a micron resolution. These are the ommatidia, which they see from, and you'll see the nerve tracts going to the optic tectum over here. This is the GI tract. Then we have segmented individual gut epithelial cells that will someday generate its own geometry of geometric characteristics. These are the embryos in the background here, with a lot of yolk inside. And as you'll see in a minute, we can actually see what are called setae of the feeder plates. These are the feeder plates here, little fingers that pull the food into the mouth. These are fat cells whose size is coincidentally about the size of a cancer cell. They're massively polyploid, which is something I didn't even know occurred in these things, and which makes them cells bigger. And these are the cross striations analogous to those that you saw earlier in the zebrafish air bladder. And here's the individual fat cells that we've segmented one just to illustrate its substructure. So every time I look at this movie or this organism, and there are other ways to look at it, I actually learn something else, or have more questions, actually, mostly. All right, so, is this, I think this is the same one, right? Yeah, I think we'll just We had another movie that shows actually octopus. Let me see whether I got that or not. Nope, it's not in here. I made a mistake and I didn't include that. So anyway, there's this is a, more than a decade long project involving a lot of people. Steve designed the camera. Patrick is the physicist in the University of Chicago who introduced me to the synchrotron. I didn't even know exactly what a synchrotron was until he introduced me. Phil's in his lab. We got multiple people I've been working with in machine learning. There are projects I didn't get to talk about. There are people at the synchrotron, the Department of Energy, at Berkeley, and at Argonne National Lab, who have been key in helping us. Michael Moorhead's here, who we're showing virtual reality pictures of our fish at a meeting. They just dropped everything and came to an international zebrafish meeting some years ago. David Mendrell helped with the engineering. Casey Ang. I mean, actually, I can't possibly have done anything that I showed you without a large number of people who have been in my lab currently and in the past. And I have a lot of funding from NIH, and I can't thank the Gitlin enough for past funding and present funding. And I don't know, just a lot of gratitude to this institution and the people in it. This is for fun, and I'll be open for questions. Let's see here. It's now. Oh, I ended. I wanted to leave a lot of time for questions. So, does anybody have any questions? This is a fun experiment. I asked a past postdoc 10 years ago, can you cut a hole in this thing and have us walk through it? So there's the eyes and the brain showing up here. Optic nerve tracts and going to the optic tectum. Gills, you can see the gill leaves here. This is the heart with blood cells in it. This is the gut, and this is the edges of cut surfaces of teeth, and then that's the air bladder above, and the holes are like the goblet cells, and it's ending with this pancreas. These dense cells have zymogen granules in it, and this is the liver. So I think I better stop distracting you with all this. Okay, I'll just end with that.
Really impressive work, Dr. Cheng. Thank you so much. Does anyone on the participant list have any questions they'd like to ask in chat or verbally? Just feel free to unmute.
Tougher the question, the better. Since I'm about to submit my grant, I better cover all that. Oh, I wonder what happened to that one slide. Let me see here. Oh yeah, I guess. Good enough.
All right, it looks like we have a quiet group, so thank you all for attending today's session. We do have an upcoming translational science seminar on June 22nd. It's Dr. Natalie Kirby who will be presenting on reducing risk of bias in study design and reporting in human physiology research. So we look forward to having you, and please keep an eye on our CTSI newsletter for more events coming up. Thank you, Dr. Cheng.
Okay, you're welcome. So again, anybody who's even remotely interested in imaging their samples from their whatever model system they use, or whether it's human samples, and we are looking at human tissues, melanoma, skin lesions, any kind of tissue that you think three-dimensional information could be helpful to you. We're collecting collaborators now who have had images of theirs from their samples, whether it's an organism. Frankly, for fun, we've looked at sequoia wood, Arabidopsis plants, and mouse embryos, frogs, axolotls. So there's a lot of people talking to us about imaging their samples across the globe right now.
Great.
I'd really like to include a lot of people from Hershey and Penn State University.
Wonderful.
All right.
Thank you very much. Have a good one.
Thank you.